This is the first paper that addresses technical problems when sequencing small RNAs derived from extracellular vesicles from cells. NGS samples require stringent preparation and quality control. Due to the nature of EVs, the QC process of EV samples deviates from the norm. The advantage of this protocol is the QC steps therein for first-time users. Demonstrating the procedure will be Junyi, a research assistant from my laboratory.
To begin, plate human mesenchymal stem cells at 2,000 cells per square centimeter in fresh MSC media in 5 T175 flasks. Then, grow the cells at 37 degrees Celsius in 5% carbon dioxide environment and replace the media every 2 to 3 days until 5 flasks of 90% confluent cells are obtained. Next, wash the cells 3 times with 20 milliliters of PBS per flask. Add 20 milliliters of EV collection media into each flask and incubate them at 37 degrees Celsius in 5% carbon dioxide environment for 48 hours. Collect the media and centrifuge it for 10 minutes at 300 G and 4 degrees Celsius. Then, collect the supernatant and centrifuge the media for 20 minutes at 2000 G and 4 degrees Celsius. Collect the supernatant again, and centrifuge the media for 30 minutes at 15,500 G and 4 degrees Celsius. Collect the supernatant for the last time. Next, transfer the media to ultracentrifuge tubes. A 60 Ti fixed-angle rotor is used here, and